Can everyone hear me okay?
Yes. Good morning.
Good morning. I got a weird Zoom message just now saying that my speaker isn't working properly and that the recording won't include audio. So I'm a little nervous about that. I think I don't want to end this. Well, you guys can hear me. So that means that the speaker is working. So I guess I will trust that and just go forward with that, and hopefully our recording isn't messed up. But I guess we shall see. Let's see here. Let me pull up. While I pull up today's PowerPoint slides, does anyone have any questions about anything? Give you a second to think about it. You guys had lab yesterday. I spoke with Bruna and it sounds like things went well. Just so you know, going forward, there will be more plants available depending on what type of plants we're looking at. It's just with the bryophytes, it's a little tricky sometimes to get material, and also they're very small, but hopefully there was enough that you guys can like kind of pull them apart and look at them up close. And obviously there's a lot of preserved specimens for the bryophytes, so that's nice too. I pull up the lectures. Wow. I have a question in the chat box. Right, you guys had an assignment due before class this morning. Let me just pull up the PowerPoint while I have it up. And I have a question in the chat box. Will the assignment one's answers be posted? Absolutely, they will be posted. I usually wait a second or sometimes, so you're supposed to turn in the assignments on time, but sometimes students forget to turn in the assignment, and so they want to still turn in the assignment for credit, which I let you turn in the assignment late up to 2 days. So I don't post the answers until after that period of time, so that late people who are turning in the assignment late don't have access to the answers until after that. Yep, so let's see, today is the first. If the assignment answers are not showing by, say, like after class on Thursday, please send me an email to remind me to make the answers available. All right, let me share my screen. Let's see here. Okay, so make this small. I can't, this is already our fifth meeting in lecture, which is, gotta mute that, which is pretty incredible. I can't believe how fast these semesters seem to fly by. Which is why it's important to stay on top of the ball, which is why I give you guys those assignments to make sure you're going through the material in pieces rather than trying to cram, because as I told you at the beginning of this semester, it's very impossible to cram all this material. So hence the regular assignments. Let's see here, just check. Great. Another chat box. Michelle's asking, was there a discussion post for the last lecture? So what I did for the last lecture, it took us 2 meetings to cover lecture 3. And so I just made a single post, a single thread for lecture 3. You don't need to post in there twice. You could just post in there once and it will count. Yeah, it would have been too confusing to have 2 separate discussions for 1 lecture. So there's just the one. You only need to post 1 to get full participation points. Cool. Was there anything else? First day of Black History Month. That's one thing I want to mention today. I'm going to reach out to some of my friends at Black Botanist Week, which is a thread you should follow on social media if you don't, because it's got a lot of cool people there, to see if I do special guest lectures sometimes in the course. Last semester, I did them toward the end of the course. This semester, I'm thinking about peppering them in like throughout the course. But in honor of Black History Month, I'm gonna try to incorporate that into sometime this month. The other thing I wanted to mention is, before I go on and give you time to think of if you have any questions, a lot of students take this course obviously because they need it to graduate. But a lot of students also end up doing very well in this course. And so toward the end of the semester, I usually get lots of emails from students who want me to write letters of recommendation. I'm happy if you're a hardworking student and you're doing well in my class, I'm happy to write letters. I've written many letters of recommendation over the year for students. So I'm putting that out there because I know how important it is to get those letters. Let's put it this way, every job that I apply to right now, I need letters of recommendation too. So it's the gift that keeps on giving. So I'm happy to write letters for people who do well in the course and who want letters. The other thing though I would say is it's hard to get to know you one-on-one in this format, right? And so what I would encourage, if you are planning toward the end of the semester to ask me to write letters, I would recommend that you email me within the next couple of weeks and we set up a one-on-one meeting so that we can chit chat. I can start to get to know you a little bit better. That way, when it comes time to write a letter, I can write a more compelling letter, because many people end up doing well in this course. The majority of students get A's and B's. So just getting an A in the course, it's harder for me to write a compelling letter because, so yeah. So I'm just making note of that if that's something you're planning on doing. Okay, any other things? Yes, so this is our 5th meeting and we've already covered a good amount of material. We're going to keep on going, cover more material. And I just want to say, like I mentioned, a lot of students need this course to graduate. So many of you are doing really well already and keeping up with the work. Keep that up, right? If you need this course to graduate, act like it. And a lot of you are doing really well. Like I said, lab yesterday sounds like it went well. So I'm happy. I'm really happy about that. But stay on the ball, right? Stay focused, because it's a lot of things. For the audio mic recording, you can check if the system computer audio is the same on your device and Zoom. Well, we'll have to see. We'll have to see what happens. Because I'm not gonna mess with it now. So fingers crossed, no matter what, at least the slides will be available. Okay. So we're going to finish up the bryophytes hopefully today. We're going to look at liverworts and hornworts. Last time we looked at the mosses, right? True mosses and peat mosses. But that's a good tip to have here. Let's see. All right. Let me just mute Henry. Okay. All right. So yeah, so this is our 5th meeting. Now I have all these boxes up. This is crazy. So you had an assignment due this morning at 10 a.m. As we talked about last time and the time before. That assignment covered the first 2 lectures. Hopefully you found that assignment pretty painless. The assignments are not meant to be hard. But that was due this morning before class. If you didn't turn it in this morning before 10, send me an email so that I can reopen the assignment for you on Canvas. Just a reminder, as stated in the syllabus, you need to turn in your assignments on time. Late assignments are automatically dropped a letter grade, and I don't accept assignments that are 2 days late. So make sure you turn these in on time. Assignments collectively are worth more than one exam. So they do matter. They're also a way for you to have an opportunity to see what test questions might look like. They're to help boost your grade because they're not meant to be difficult. They also let me know who's keeping up with the material and who needs to put a little more effort in. All right, so enough of that. last time we covered the part of the bryophytes, the non-vascular plants. We covered those true mosses, those peat mosses. Today, I'm going to finish up the bryophyte with the liverworts and hornworts, and get ready to practice saying these words, right? We have the phylum Marcantiophyta and the phylum Anthocerophyta. And just like I said last time, as a reminder, when you see this ending on a word, phyta with an A, right? You know you're talking about a phylum. So the phylum for the hornworts is Anthocerophyta. The phylum for the liverworts is Marcantiophyta. And as we learned last time, the phylum for the mosses is Bryophyta, right? So if it ends in phyta, you're talking about a phylum. Not to be confused with bryophytes, which just refers to all of them, right? All the non-vascular plants. Okay. Where are we in the plant kingdom? Well, we're still very much at the beginning, right? We're only into those non-vascular plants. Here is a little picture again. We saw this picture before during our last two meetings of what these little guys look like as a cartoon. But first, we're going to talk about those liverworts. Liverworts are, the word liverwort is weird. It sounds like some Harry Potter word or something, right? Liverwort. Why is it called that? Well, it's called that because whoever described them first thought they looked like a liver. They didn't have an internet and cable back then, guys. So they had to work with what they had. But yeah, they thought it looked like a liver. AI, no. Well, you'll see it on the cartoon. Somebody, Hala said in the chat, how? In the cartoon, it may not look so livery, but the idea is that this thing kind of looks livery. It might look more livery on the next, in a real picture. But yeah, so liver, because the gametophyte has a shape that, another word we've seen before, a thallus. Hala says that she sees a silly face. Yeah, I kind of like see it too, right? Like, there's the eyes, maybe a little. No, well, it's not called silly face wort. It's called liverwort, because the gametophyte has this liver-shaped thallus. Thallus was a term we learned last time. Thallus just means a flat green thing, as versus a leafy green thing, right? A flat green thing. And then wort comes from this Anglo-Saxon word, which means herb or plant. So if you were to break up the words there, you have liver plant, liver plant. Poor liverworts. I like silly face plant much better. Okay. Here's what they look like in real, though. Here's what they look like in real. So let me just move the chat box. As always, I like to give you guys some tips or help about how to pronounce these long, brand new words that you've never seen before, right? So the phylum Marcantiophyta, right? Marcantiophyta, liverworts. In this picture, the liverworts are kind of like these green ribbony things. I'm gonna draw a square, look, or a rectangle. Inside of there is a bunch of green ribbony things, right? Over here, all these things, green ribbony looking dudes. Those are liverworts. This slide, since we learned it last time, also contains some green fluffies that are those sphagnum mosses mixed in here with the liverworts. So just so you don't get confused, the things that are in the circles are not liverworts. The things in the circles are peat mosses or sphagnum. But a lot of times in nature, when you find moss or liverwort or hornwort, a lot of times in nature, they like to grow together. So it's not uncommon to see something like this, where you have a big dense mat of liverworts, and then they're mixed in there are some mosses or other things, maybe even some hornworts. All right, liverworts are really old, based on fossil record and molecular data. They showed up around 375 million years ago, give or take. There's currently about 8,000, give or take, species known as similar to the mosses. There's probably at least another 8,000 out there, if not more, that we haven't described yet. And because we are talking about non-vascular plants, right? Non-vascular plants. We know that the gametophyte generation is dominant. Because it's a non-vascular plant, we already know that. Right, like on this previous slide, the green flat ribbons, which are the liverworts, and the green fluffy things, which are the sphagnum moss or peat moss, are the gametophyte generation, because that's the dominant generation in the non-vascular plant. So we know that already. There's two types that are basically described by their shape. So they're those thyloid liverworts. Again, don't get scared of the word thyloid. It just means a flat green thing. Thallus is a thallus, right in the chat box. Thallus is a flat green thing. And if something has a thallus shape, we say that it is thyloid. That's what we do. So there's the thyloid liverworts, or the flat green liverworts, of which there's about 1,600 species. And by far the most common type of liverwort, grows in a lot of different places. Example genera are Connocephalum and Marchantia. Marchantia we will see in lab. And what's nice about Marchantia is the genus name comes from the phylum name. So you have to remember less things. You have the phylum Marchantiophyta, and one of the genera, that's the plural, one of the genera you're going to look at in lab is Marchantia. So that's nice. There's also the leafy liverworts, of which there's a lot more species. We don't really focus on the leafy ones too much in this course because, number one, they kind of look like mosses to the untrained eye. So that can be a little confusing for people who are just starting to kind of learn plants. So that's one reason you don't focus on them. But the other reason we focus more on the thyloid liverworts are because they are by far the most common and widespread. So if you're hiking around in the woods in New Jersey or New York, you can come upon them. Whereas the leafy ones, not as much. They'll look more like a moth. Oh yeah, and this is a reminder from our very first class, because I like to keep you guys keep things up. Remember that when you see something that's italicized, right, like these words here, or underlined, which you probably will see in the lab, followed by this SP. SP just stands for species. And remember, the underlined thing is the genus name. So for example, for Marchantia here, this is just saying all of the species in the genus Marchantia, all of the species in the genus Connocephalum. A question in the chat box. Can it also be not italicized or underlined? No. So if you're going to be correct, anytime you write a genus name, so genus, anytime you write a genus name or the entire species name of a plant or an animal for that matter, it needs to be italicized or underlined. So just for that reminder, for humans, for example, you have like Homo sapiens. Whoops, sapiens. So if you were to write Homo sapiens, for example, those two words would need to be underlined or italicized. If you wanted to talk about all of the species that are in the genus Homo, you could write Homo sp italicized or underlined. If that seems unfamiliar to you, go back to the first lecture slides. There's good material there for that. Okay, let's move on. So leafy versus thyloid liverworts. When you look at them, it's pretty easy to tell them apart, right? The ones on the left here, the thyloid shaped ones, flat and green. Flat and green almost looks kind of like maybe a seaweed, or maybe even, I don't know if you know what a lichen is, but almost like a lichen. Flat green thing. These are the ones we'll focus on in our class. Thyloid. But there's also the leafy ones. And like I mentioned before, see the leafy ones here, this is Perella on the right. Looks kind of like a moss, like a little bit mossy. So that's why we don't focus on them as much, because they're just not as conspicuous. Where do you find liverworts? Lots of places. But most of the time they like the kind of tropical or temperate environments. Remember that just like all the other bryophytes, right? Just like all the other non-vascular plants, liverworts require water for sexual reproduction to occur because of the sperm swim. So because of that, most of the time, most species will live in places where there's ample water, like tropical or temperate environments. But there are some species, much like the mosses, that can live in very extreme environments. One thing also similar to the mosses that we learned about last classes, liverworts are also those type of plants you can say is early successional, meaning they can grow on bare soil or rock, as shown in these pictures here, right? In both these pictures, this is liverworts growing on rock. Remember I said when we were learning about the mosses, what does that mean, early successional, right? Well, like for example, if there's a new island that shows up via volcanic activity, and now in the middle of the ocean, you have this bare rock island that just showed up. Well, liverworts, mosses, and hornworts, those are examples of plants that might be the first plants to show up on that new habitat. Another example I mentioned in previous classes is after a very severe wildfire, or even after a mudslide, or something like that, where the landscape gets, all the vegetation and everything gets removed from the landscape. And so now that's a new habitat to go on to. And so liverworts might also show up there first, which is early successional. Another thing that liverworts are, another word that we've already encountered in this course, liverworts can also grow as what's called epiphytes. So we saw this term when we learned about the non-vascular plants in general, but epiphytes means that you are a plant that can grow on another plant, right? Usually trees or shrubs because they're woody. But here's like an example of some leafy liverworts that are growing on a tree. And you can see how mossy they look, which is why we don't really focus on those. Here's an example of a thyloid-shaped liverwort growing as an epiphyte. So this is another one growing on a tree. A lot of species of liverwort are aquatic, and so they either float on or live under the water, usually fresh water. If anyone here, so I have a couple fish tanks. I'm like big into fish tanks. I think it's like really fun activity. I started a few years ago during grad school, and only have freshwater tanks. But I found it really interesting when I started to get into the hobby to find out that a lot of the plants that are sold at aquarium stores or fish stores, they're actually liverworts. So if you also are into keeping freshwater fish tanks and you have living plants in there, you may be surprised to know that you already own some liverworts in your house. Yeah. So here's an example here of, see, Mara, I think Mara is how we pronounce that name. Let me know if I'm saying it wrong. Mara has a fish tank. Yes. Okay, good. Thank you, Mara. Yeah, so that was something interesting. Even me as a plant person, when I first started keeping a fish tank and buying plants for it, I had no idea that they don't tell you that at the fish store. Sometimes they are called a liverwort, but I thought that was pretty cool. Yeah, so here's an example of them living floating on water. Here's an example of them living submerged under the water. Hala says, they look like plant bugs. These do, you're right. They're not, though. They're not. Now you might be thinking, oh, I have to go to like the tropics or some faraway place to look at liverworts. You are wrong, kids, because some species can even live in urban environments like Newark, New Jersey. So I took this photo. This is a photo I took on campus in Newark a few years back, right outside of so this class, when it is in person, like the lecture component, it's always in the Center for Urban and Public Service because that's the only building that has a room big enough to have 40 students in it at that time. Anyways, so there was one day I was leaving class. I don't think I was talking about liverworts that day, which would have been very ironic, but no, one day I was leaving class, walking away from the building, I looked down and whammo, under a rain gutter right there in Newark, New Jersey, liverworts growing on the bricks. So look down, look down sometimes because they could be in the city too. Okay, so here liverworts are, all right, so first we're going to talk about the gametophyte generation first. Gametophyte generation first. And just because I like to really hit this home, what is a gametophyte? A gametophyte is a phyte. You can say it with me, right? This is a phyte or a plant that makes what? Gametes.
Gametes.
Yay, gametes, egg, and sperm. That's right, gametes, egg, and sperm. Liverwort gametophyte plants are small, right? Very small. Here's a dime. In case you've never used cash, kids, I know your Venmos and your Cash App and all that. In case you've never used actual money, this is called a dime. And you have to trust me, they're small. They're small. I'm making a joke. But yeah. This is a dime. This is what we used to use in the old days. Piece of metal that we would pay anyways. Yeah, so just to give you an idea of how small gametophyte plants are, right? There's one right there, super small. And so definitely very, very small, very, very small. As we go forward though, I'm not even gonna say that. I'm not gonna say, okay. So yeah, gametophytes are small. Gametophytes are non-vascular because we're in the bryophytes, right? So we know that they have no true roots, they have no true stems, they have no true leaves, they don't have any vascular tissue. We already know that. And we know already now there's two types, thyloid or leafy, in terms of gametophytes. All right, this growth by apical meristematic cells. So just because this keeps showing up in the lecture, I should mention, so like meristematic looks scary. Maybe since you guys are fourth year biology students, you may have heard the term meristem before. If you haven't heard the term meristem before, that's okay. But maybe you've heard the term stem cell, like a stem cell. Well, meristems are like stem cells, in that they are cells that can divide and grow into, yeah, stem cell, exactly. Like other stem cells, meristems are cells that can grow into all different types of tissue. So for example, leaf-like tissue, stem-like tissue, and later once we get into the once we get into the vascular plants, then it will be actual leaves, actual stems. But the point of this, I just want to show you what this means. Right, Mara says they are capable of cell division. Correct. They are, and they can give rise to all different types of plant body parts too, just like a stem cell. The reason I'm showing you this slide here, though, is to emphasize the simplicity of a liverwort gametophyte body. Very, very simple. So remember, we're going to focus on the thyloid ones, but even if you talk about the leafy ones, what's the best color to use here? I guess yellow. Very, very simple body plan in that you have a thing growing here. Let's see, look right here. Let's do this. You have, like, let's say you have a tip growing, right? Like this tip right here, growing via mitosis and cytokinesis. And what happens as it grows is it's going to split into these kind of equal branches, something that we call equal dichotomy. All that means is it grows very simply, and it basically is a series of splits, grow, split, as the plant body grows. So for example, on this thyloid one here on the left, you see here, grow, and then it splits here. Eventually, if it keeps growing, this could even split again here, and so on. That's a type of growth pattern that we call equal dichotomy, right? You can see here, grow and then split. Down here, look, grow, split, and then, like I said, it even goes further here to grow and then split again. So grow, split, grow, split, grow, split. On this leafy one here, you can see here, oops, wrong way. Right, grow and then split. Meristematic cells will split, divide, and have this kind of equal dichotomy pattern. See, grow and then split. The reason I mention this is because this is just a very simple body design, a very simple body plan. As we move up through the different phyla of the plant kingdom, the plant body becomes a lot more complicated. So I'm just pointing out the simplicity as it is here. Another thing about the gametophyte, similar to the mosses, is there's things called rhizoids. Just remember, rhizoids are not roots. They are little projections that help the gametophyte plant attach to things or anchor onto something. In this drawing here, you see the green flat gametophyte. So like here's the green flat gametophyte. And all of these kind of hair-like things are those rhizoids.
So with the ones that were in the water, the black things that were going into the water are rhizoids too, right?
Oh, those little projections coming off.
Yeah, the ones that look like legs on a bug.
Yeah, that's a good point. Probably, but for those, Henry, those are a little unique in that those ones that look kind of like the bugs, they don't attach to anything, they float. And so in that, here, let's go back just to take a peek at that slide real quick. Those float. Those are actually probably just parts of the, see here? Yeah, so those are probably not rhizoids because those are probably just more leaf-like things kind of hanging off of them. We could talk more about when we get to a little bit higher evolved. What probably happened is here, just for, so here's your flat green thing. And then you see all these little like leg looking things below. We don't have time to go into it today, but suffice it to say that those were also probably at one point flat green things as well. But because they're going to be submerged under the water over evolutionary time, they just turn into kind of these little projections. Because these float, these don't. Okay, good. Yeah, these float, so they don't need to anchor to anything. And Whereas rhizoid's main purpose is to actually anchor the plant to like a rock, for example. So the floating liverworts don't really need rhizoids. Yeah, but just keep in mind with these rhizoids that they are not roots and very thin. So they're usually only one cell. That's why it says unicellular, one cell thick, very poor at absorption. Main job is to hold them to things. Okay, let's talk a little bit about what's going on inside of the liverwort gametophyte plant body. So when we learned about the mosses, we learned the inside of a moss gametophyte body had like hydroids, leptoids, and parenchyma tissue. And that's kind of all we really talked about inside of those moss gametophyte plant bodies, right? In the liverwort gametophyte, the plant body is a little bit more complex at a cellular level than, say, the mosses. Let's see. So how are they complex? I guess I'll use black. Black might work well on this slide. See, so it says thyloid gametophytes are complex. What do I mean by complex? Well, for example, they have different layers of cells that allow them to do different things. So for example, and just to give you orientation, here's the rhizoids, right? These are the rhizoids at the bottom of the gametophyte plant. On the top, or dorsal, see this is dorsal? The way I remember dorsal is top is I think of a shark has a dorsal fin, right? And it's on like the top of a shark. So on the top or dorsal surface of the flat or thyloid shaped liverwort gametophyte plants, you have this photosynthetic layer. So you can see here, it's all green. And that layer is going to perform photosynthesis via all the different chloroplasts. So that's one layer of complexity. Underneath, another layer of complexity, which we will see better on subsequent slides, is that there's pores. This is this is not a good looking pore. We'll see pores better on the next slides, but trust me, there are air chambers and pores in the liverwort gametophyte plant body that help with gas exchange, which we did not see that. We did not see that in the mosses. And then there's a lower layer in the gametophyte plant that serves to store food in the form of starch. So this upper layer here makes the food, and then that food is stored in the lower layer as starch. So this is a cartoon. This is a real microscope slide. Show you the same things that I just discussed. So here are those rhizoids again at the bottom, so you get oriented. Here is the top surface, right? Look, the dorsal fin of a shark. Here's the top surface. Look at all of this green stuff. Green stuff here. Well, I don't know about Hala saying I got a picture from yesterday lab. I have the same one. Well, you don't know if you looked at liverwort slides yesterday, but may have been something similar in moss. But let's focus right now on this thing here. So top layer is those photosynthetic layer that contains chloroplasts. That's where photosynthesis is going to happen. That's where sugar is going to be made. Yeah, it probably looked similar, Hala. And then down here, this layer is where the food or the products of photosynthesis, sugar, is stored as starch. And if you look at them under the microscope slide, starch stains purple. So all these purple dots throughout the slide are those starch grains. And then I said there's air chambers and pores. So that's what this is showing you here. This is a air chamber, and then this opening here is the pore, which is something new that we did not see in the mosses. these air chambers and pores. Okay. These air chambers and pores help with gas exchange. They let oxygen gas flow out and they let CO2 gas flow in. Remember, you need CO2 gas in order to perform photosynthesis, which we'll talk more about photosynthesis in a later lecture. One thing though, so this is a new structure and this is kind of cool. I mean, you could it's a lot easier for CO2 gas to go in. It's a lot easier for oxygen gas to flow out. One problem though, in terms of, we're still talking about a pretty simple plant, is that this gas exchange is not regulated. So the pores are just open all the time, which may not be great. It might be great if the environment is, nice, humid, kind of environment. But if you're in a more extreme environment, it means that you can dry out a lot faster. You can think about it as when you think of a pore, right? You can think of a window with like no glass. No glass. It's just an open window and you can't close it and it's just open all the time. Later, we'll learn about gas exchange in a different way where it's more regulated. But liverworts are super simple, so they don't have that yet. This is another microscope slide showing you cross-section through the one of those thyloid gametophytes again. So just to keep you with what we've been, look, that green layer, photosynthetic cells, this is the layer that stores starch underneath it. Rhizoids would be coming out from there. And so we're talking about gametophyte, right? Plant that makes gametes, egg, and sperm. So we know there has to be sex organs on this gametophyte plant. And those sex organs are going to be coming out of the dorsal side. So one thing we just learned that liverworts have that mosses didn't is these pores, right? It's one difference in addition to being flat, right? So flat is one difference, a little bit more complex in terms of the different cell layers. That's another difference. Pores, right? That's another difference compared to the mosses. And then this is another difference, and maybe one of the most important differences is that there on the liverworts, there are special structures that elevate or lift up the sex organs that the mosses did not have. And here they are, So we're gonna learn a little bit more these roots here. So liverworts have special structures that lift up or elevate the sex organs. Those special structures are called antheridia 4. Here you go here, right? Antheridia 4 and archegonia 4. Just so you know, and it'll say this on the slide going forward, I'm pretty sure, but 4 means to bear or carry. I'll write it in the 4 equals to bear or carry. So if you know that 4 means to bear or carry, so an antheridia 4 is a structure that bears or carries antheridia. And if you're an archegonia 4, you're a structure that bears or carries archegonia. So these new words, antheridia 4 and archegonia 4, those are not the sex organs. They are these structures that elevate the sex organs away from the flat green thing. So if you look here at these old, ancient drawings that are so old, maybe even 100 years old, I have to look. But these drawings show you, so this whole thing that's coming up, That's an antheridia 4. And this whole thing that's coming up, that's an archegonia 4. On the top here, you're going to have antheridia. And up here, and we'll see better on a, don't worry about sporophytes right now. On a future slide coming up, you'll see better. But up here on this structure will be archegonia. And just as a reminder, we've covered it a lot of times, but antheridia, those are those male sex organs that are going to release sperm. Archegonia is the female sex organs that are going to release, or no, that are going to make egg, but not release egg. Egg stays. Okay. Just when you're looking at the slides later, these nice circles will be there, or ovals. All right, here's a question for you guys. Look, you have a male gametophyte plant and a female gametophyte plant, which means that you have 2 plant individuals that each have one type of sex organ on them. 2 plant individuals that have one sex organ or the other sex organ on them. What is it called? What is it called when that's the case? I'll wait. Two plant individuals or 2 houses. No, everyone's asleep.
I'm sorry, can you repeat the question again?
Yeah, so you have 2 plant individuals here, right? One plant individual that has male sex organs on it, one plant individual that has female sex organs on it. What do we call that when you have 2 individuals? Yeah, see, Geetika has it in the chat, right? Dioecious. Thank you, Geetika, right? Yes. Is that two individuals, each with one type of sex organ? Or you could say 2 individuals, or you could think in your head 2 houses, right? 2 houses, 2 plants. So thank you. Thank you. Okay. And oh yeah, this is just so when you're looking at the slides later, right? 4 is the bearer of, or the structure that bears or carries something. All right. Here is, so yeah, so if there's a, so if it, yeah, so if there was a single gametophyte plant that had both sex organs on it, that would be monoecious. How, yes. Yep. Or one house, right? One house. All right, so these were some really old drawings showing the antheridia 4 and the archegonia 4. Here are some other not as old drawings, but still kind of old. And I know it looks a little fuzzy here, but it still is good because it can show you where the sex organs are. So here's the top of a antheridia 4. I'm gonna have to mute Kuma there. So here's the top of an antheridia 4. Here's the top of an archegonia 4. The reason why I'm showing you that is because just showing you here in the antheridium 4, the antheridia, or the organs that make sperm, are going to be on the top side of the antheridia 4. And that's because, remember my amigos analogy, right? Because we're talking about the non-vascular plants, they need sperm, they need water to let the sperm swim. So like a raindrop's going to drop on top of this antheridia 4. So therefore the organs that make sperm need to be on that side of the surface, right? Because the sperm's going to need to like splash out with the raindrops so it can swim away. On the other hand, if you're looking at the top of an archegonia 4, the archegonia are actually going to be on the underside. And what will happen, we'll see this on a slide coming up, but let's say a raindrop drops on top of this antheridia 4, allows the sperm inside of here to like splash out. Whoa. And let's say that sperm lands, or the water drop lands on this antheridia 4, sorry, archegonia 4. Raindrop lands on the antheridia 4, splashing sperm out of the antheridia, right? Here's sperm splashing on from the antheridia, woo, onto the archegonia 4. And what'll happen is the water will kind of go around the arms of these like that, because of the properties of water. That's what'll happen, and allows the sperm to swim to the egg. This will be shown on a slide coming up too. I'm just showing you it first time here. All right, here's what they look like in real. And in lab, most likely in lab, you'll see preserved specimens of these, which I call pickles, because it's basically like pieces of plants that are in jars. So you'll see pickles of these in lab, because they're hard to find in nature. Even me, in looking in them in different, even in the city and in the woods, I've only seen these on plants a few times. So it's hard to find them living for lab. So we have preserved specimens for lab. Here are those antheridia 4s. If I was in this previous here, remember that's this one here. This one here that has the antheridia on the top surface. So why? Because rain needs to fall, rain needs to drop on top of these, right? Splash out the sperm. Look, oh, boom, from rain, boom, splashing on you. And then like, oh, I'm sperm, yay, I can like swim out now and go to the egg. Ooh. How do you tell the antheridia 4 and archegonia 4 apart? They're similar in structure, but if you start to look at them closely, you can see little differences. So one is, let's see, what's a good color on here? I guess white. Here's one antheridia 4. Kind of looks like an umbrella. That's how I think about it. You can see here, kind of looks like an umbrella. There's a little stalk holding it up. That's how I think of them, is that they kind of look like an umbrella. Some people think they might look like a mushroom. Right, this is them again. Switch back to black. Right, kind of looks like a little umbrella or a mushroomy thing. On the top here, you have a cross section through microscope slide. So this is the top part. Here's the little stalky thing holding it up. And then each of these darker circles are antheridia. And then if we were to zoom in on one antheridium, this would actually say antheridium because we're looking at one of them. But this one antheridium is full of cells that are going to make sperm, or you can say full of cells that are going to generate sperm, or spermatogenous, right? Remember that word, genus. I got a question from Hawa. I know last lecture you said the water is the only way sperm can swim to the egg. It sounds silly, but can wind also make sperm travel too, or no? Or no, only water. So in the that's a good question. It's a good question because you would think that's a good question, because look, if wind is blowing here, right, you can see maybe it could like spread some sperm out. But no, in the non-vascular plants or the bryophytes, they need only water, or maybe some other liquid, but only water is going to allow the sperm to swim. So in the bryophytes, wind does not move sperm. Wind does not move sperm in the bryophytes. Yeah, because remember, sperm swim, sperm don't fly, right? Like sperm don't fly, sperm swim. So sperm need water to swim in. Later, what does fly though is the spore that the sporophyte makes. That does fly in the wind, but the sperm does not fly in the wind. The sperm doesn't fly, the sperm only swims. Yeah, right. Henry's saying that the wind will affect the way that the water splashes, but yeah, so sperm swims, not flies. Okay. So this was the antheridia 4s, and just get to know them a little closer up, right? And then you have the archegonia 4s. So I say that the antheridia 4s look like little umbrellas, And I say that the archegoniophores look like either a palm tree or an octopus. So here's the top of an archegoniophore. Here's another top of an archegoniophore. And as you can see, you can't see the stalk so well, but just know that there's a stalk right here. And then on top of the stalk, there's like these weird arms kind of like hanging down. Sorry, I'm not drawing them super well. Right, look, here's some weird arms like hanging down. So if I were to draw like side by side here, I'll do it over here, what an antheridiophore looks like as versus an archegoniophore. So like here you go, here's antheridiophore. Looks kind of like an umbrella. Here's archegoniophore. Looks kind of like a palm tree or an octopus, if I had to compare the two. Okay, so here is archegoniophore. So this was the real, right? This is real again on the left. Here, archegoniophores, right? Look, palm tree looking thing. Here's the stalk. Here's like little arms coming off of the palm tree, right? If we were to look at it under the microscope, a lot of times when you look at it under the microscope, you can only see two of those arms, right? Because you're doing a cross section through the thing. So here's one arm, here's another arm. As I showed you on that drawing a few slides back, unlike the antheridiophore, the archegoniophore has the sex organs on the bottom, right? So on the antheridiophore, they were up here because of the raindrop thing. Remember I said before that the archegonia are going to be on the underside of that archegoniophore. And if you were to look at an archegonium up closely, it would look very similar to what we've already looked at in terms of an archegonium, right? It has the same parts. You have that neck, here's the neck. You have a swollen part called a venter. And then inside the venter, you have the egg. And this is a cool slide here because this actually shows this is a sperm arriving to fertilize the egg. Very cool. Okay. I'm going to, in the interest of time, because you guys are going to see this in lab on Monday, I'm going to go through alternation of generations with a liverwort. And then we will break for the day and we will deal with hornworts on Thursday. So this slide is an overview slide of alternation of generations in Marcantiophyta, liverworts. We'll go through this parts of this slide together here, but just so you know, this is the whole thing on one slide, right? Just to get you oriented, like you got sporophyte plant here, and then gametophyte plants over here, right? So this is the whole thing on one slide. This is from your book. Okay, so we will start with, let's see, we'll start with the spores. So we know sporophyte, right, is a phyte that makes spores. We know that. So here's the spores that they made. And these is what I was saying before. Spores are vectored by the wind. So yes, water is needed for the sperm to swim because sperms swim, but spores up here, spores fly. Spores fly in the wind. So that's where wind comes in. All right, so there's spores. Remember, the spores are the result. A lot of this is going to start to, you're going to be like, ah, this is where she was saying, don't be worried about these words because they keep repeating over and over again. Here's spore. Here's another spore. Whoops. Here's another spore up here. Sorry, all my Zoom features are getting in the way of me going up on that part of the slide. Spores are what? They're haploid because they were made by the sporophyte plant via meiosis, right? Cytokinesis. So we're already talking about haploid. Notice in these slides that yellow color And don't worry about, yeah, gray up here, but yellow usually refers to the haploid. So spore is released from the sporophyte plant. Spore is haploid. Spore is going to grow via mitosis and grow and grow and grow into a gametophyte plant. In this case here, it's a female gametophyte plant. The other spore up here does the same thing, grow, grow, grow via mitosis into a male gametophyte plant. Well, we have female and male gametophyte plant, right? Gametophyte, right? Gametophyte. It's a plant that makes gametes. Gametophyte plant makes egg. Sorry, female gametophyte plant makes egg. Male gametophyte plant makes sperm. So we know they have to have sex organs on them that do that. Because we're talking about liverworts, the sex organs are located on new special structures that we just learned today. Brand new words today, right? Archegonia 4 and antheridia 4. As I said on the previous, 4 means to carry or bear, or the bearer of. So this little structure here is a structure that bears or carries archegonia. This structure here is going to bear or carry antheridia, right? Let's start with the one here. So if we were to zoom in to the top of that archegonia 4, right, we would see this under the microscope slide. Remember, I said usually when you do a microscope slide of the archegonia 4, you only see the 2 arms. So here's one. The other arm is not shown really on the slide. Archegonia 4 is what I said, looks like a little palm tree. Sex organs are going to be on the underside, right? Archegonia on the underside. If we look at the male, the antheridia 4 is going to be bearing all those antheridia, right? So if we were to look at it and look, oh, our good friend raindrop is back, boom, right? So antheridia 4 is a structure that bears, whoa. And that's why we stay muted most of the time, because of that feedback. I know sometimes it undoes, but yeah, no worries, Hala. But that's why that's a good example there of why I usually just mute everybody, because of that feedback that can happen, which is it's really weird how that happens. Anyways, antheridia 4, structure that carries antheridia, right? Raindrop drops on top. Here's the close-up of the top of that antheridia 4. All the antheridia are located on the top, right? Archegonia were located underneath. Antheridia are located on the top. They're located on the top because when our good friend raindrop drops on top of the antheridia 4, the rain can go and be inside the antheridia and splash out the sperm, which hopefully there's a archegonia 4 located not too far away. So sperm is going to splash, and the water drop, like I drew on the previous slide, because of the properties of water, the water drop is going to kind of go like this around that arm. And with it, the sperm can, so like here's a sperm. This, oh, and look, ooh, plush toy. There's going to be a plush toy one day. My new product line, plant sperm plush toys. Yeah, sperm's going to swim, and then just keep swimming until it meets an egg in an archegonia. What else should I point out here before we move on? The main thing I just want you to keep in your brain, right? A spore is haploid. Spore grows into a gametophyte plant. Gametophyte plant is haploid. Gametophyte plant has sex organs on it, has structures in this case that bear the sex organs, archegonia 4, antheridia 4, those are haploid, right? Antheridia, haploid, archegonia, haploid, sperm, haploid, egg, not shown here, haploid. So just keep that in mind, haploid, 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 until da, 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 sperm swims, boop, 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 fertilizes an egg and an archegonium. Here you go, sperm. There it is swimming. Look, there's one. Swim, 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 swim. Fertilize the egg. When that happens, look, 1N sperm, 1N egg. Fuses during fertilization. 2N zygote. Now, right, some of these things are starting to feel a little bit more familiar than when we first heard these terms. That's what I was saying before, when we first started learning these things, not to get too scared, because there's so much repetition through this class. What else do I want to point out here? Right, where was the egg? Egg was in an archegonium, right? Egg was here. Waiting, egg stays. Egg was in the archegonium. So when sperm fertilizes egg, 2N zygote is also in the archegonium, right? Because that's where the egg was. Egg stays. So this 2N zygote, that's the beginning of the new sporophyte generation, and it's going to grow out of the archegonium. Right? Okay. Here we go. We're almost done for today. Here's your 2N zygote. It's going to grow via, you have to think to yourself what grow, right? Grow via what process? Grow via mitosis. And divide, make more cells. Eventually the 2N zygote is going to be a 2N embryo, multiple cells. Embryo is going to continue growing via mitosis into a young sporophyte plant. Remember, which is growing where the egg was. So this is still inside of that archegonium. As the sporophyte plant continues to grow and grow and grow, it's actually the outside of that archegonium that becomes the calyptra. Don't worry about that though. But the point is that the sporophyte plant is growing in the archegonium, right? And the sporophyte plant is going to have tissue composed of cells that can generate what? Spores, because it's a sporophyte. It's a plant or phyte that makes what? Spores. Okay. Sporophyte plant's going to grow, get bigger and bigger. Eventually, those cells that make spores, right? Those cells that make spores, sporogenous cells or spore mother cells, which are 2N. If they're going to make spores, spores are haploid. So we know that those cells have to undergo process of meiosis, which it does. And then ultimately you end up with your mature sporophyte plant. That's this whole thing. Has some of those same words we already saw, right? A foot, which attaches the sporophyte plant to the gametophyte plant. A seta, which is a stalk that elevates the capsule. And then, for the purposes of today, sporangium, which is an organ that contains spores. And then inside here, lots and lots and lots of spores, which are now haploid because of this. process of meiosis. You might see a term here, elaters. I think that they show up on the next slide. Oh no, sorry, we have to finish. We're almost done, yeah, so now the sporophyte plant contains the spores. That's where we are here. Eventually, the spores are going to be ready to be released. But before we get there, just to give you an idea, right? I see what's the best way. Yeah, I guess I can do this. Remember, where the egg was inside of the archegonium. So when the sperm fertilizes the egg, that 2N zygote, which grows into the sporophyte plant, is still inside the archegonium. Look, this is the archegonium. This is the archegonium. This is the archegonium. And the sporophyte plant is going to grow out of it. And so by the time we got to here, look, the sporophyte plant is growing out of, this was the archegonium, because that's where the egg was. The reason I'm showing you that is because here, this is zooming back out and looking at the top of those archegonia fours, right? The things I said looked like a palm tree. And so here's one of the palm trees here, right? Like there's this little palm fronds, right? A palm tree. So that's the archegonia 4. And what I'm trying to show you here is that because the sporophytes are growing out of where the egg was, they're going to hang down, and I like to say they kind of look like coconuts. So these green things on the underside of that palm tree looking thing. So the palm tree looking thing is the archegonia 4. It's the structure on liverworts that has archegonia on it. Archegonia all have eggs inside. When the egg gets fertilized, the sporophyte plant grows down from where that was, and they hang upside down like a bat. Yeah, okay. Oh yeah, that's what I was thinking about elaters, and then we'll finish up for today. So you know, you'll see this term called elaters, and elaters are in this slide here, kind of like these lines that you might see inside of sporangium. What they do, they're similar to the peristome teeth in that we covered in mosses, in that they're sensitive to humidity. And so when the humidity changes, they kind of like move around a bit, and that helps release the spores. So this is just showing you this is a microscope slide of the cross section through a sporangium, showing you the finished spores. Well, don't worry. And then this is just showing you this is just showing you what I was trying to show you here. So this is the elaters, are these like long kind of springs almost. And all those dots surrounding that are the spores. And the elaters, here, maybe I'll use black. Here's one. Yeah, so they're just going to help the spores get released from the sporangium. Yeah. Yeah. So this is where we'll end on this slide today, and we'll pick up on this next time. But just because we finished learning about the sexual reproduction of liverworts right now, let's compare the two sporophytes and gametophyte plants real quick of the mosses. So if we're going to look at like a moss, so this is on the right here, we see a true moss, which is from the phylum Bryophyta. On the left here, we see liverwort, which is from the phylum Marcantiophyta. That's what we talked about today. The last two classes, we covered the mosses. And so for the mosses, you have this kind of fuzzy gametophyte plant. And then the sporophyte plant, which is dependent, right? You get the foot, the seta, and then the capsule, right? This is for a true moss. On the left is liverwort. Same structures, foot, seta, capsule, except the only difference is that, well, one of the main difference is that the sporophyte plant of the mosses grows up towards the sky. Sporophyte plant of the liverwort hangs upside down like a bat from the arms of the archegonia 4. And that's where we'll end for today. And we'll when we pick up on Thursday, I'll go a few slides back and kind of like work into going on in this lecture. But yep, that's all we have for today. You guys have any questions? I'm happy to answer them here. Otherwise, you can write them in the discussion board on Canvas. I did remind you guys on Canvas to please participate in those discussion board threads. That's there on Canvas. That's to give you points. You don't have to write a paragraph. You don't even have to write something really profound. Just write anything on the discussion boards so I can give you guys participation points. But you can be profound if you want to, like, you know, write something impressive, that's fine, or ask a very complex question, that's fine. You don't have to. Be simple stuff. Thank you guys for all of you. Have a great day. I see you in the chat box. Thank you so much. Does anyone have a question that they'd like me to address now? Let me stop sharing this.
Dr. Becca?
Yeah.
Do you have like office hour?
I do. So office hours, yep, it says it on the syllabus. I think it's 1 to 2 p.m. on Thursdays. But because we're meeting like in this virtual way, what I would say is if you want to meet for office hours, just send me an email so that we can so that I know for sure to confirm it, and I'll send you a different Zoom link for the office hours.
That's perfect. Thank you so much. Thank
Yep.
you.
No problem. And also, even though office hours is 1 to 2 p.m. on Thursdays, I'm flexible with that as well. So like if 1 to 2 on Thursdays aren't good for you out there, I can meet at other times too. I just need to, yep.
That will be perfect. Thank you so much, Dr. Rebecca.
No problem. Have a great day.
You too.
How about even if you participate in class, you still need to participate on discussion board.
Yes.
I guess yes. I mean, here the thing is here. I will say this: like, there's a few of you are regular participants in class, right? Hawa participates a lot. Michelle participates a lot. Kiana participates a lot, and other students, right? Probably some that have already left now, participates a lot, right? Huma a lot of times, Natasha, right? There's regular people who do participate a lot, and I noticed that. So keep that in mind. But it is easier for me to keep track. You know what? You could even just write on the discussion board, I thought I made some really good points in class today, meaning you or whoever is very active in the chats here in Zoom. Yeah, you could be like, wow, I was really impressed by all the questions I asked during class today. You know, that would count. But yeah. All right. Any other questions? Oh, where are the recorded lectures on Canvas? Okay, so Henry, there is a merged Canvas page, and it says lecture and lab on Canvas, and that's where all of the material are located. I think I posted an announcement on the separate sections that all the other materials available on the merged page. So if you go to the merged page here, let me see here if I can even just pull it up. I might need to, let's see, might need to be logged back in. No, I am here. All right, so courses, we'll go to courses. I'll go to student view. Courses. Right, okay. So let me see if I could share this for Henry. Share screen. Okay, where is my here? All right, so Henry, you should be able to see my browser now. So if you go to Canvas and you go to, and you click on the left side here, courses, you'll see a course that says 2022 SP Plant Kingdom, lecture and lab. Lecture and lab. If you click on that, this is in student view right now. This is what it'll show you. What you can do, because I put everything in announcements. So what you could do is you could click on announcements and then you have everything here. For example, lecture 3 slides and recording. So you would click on that. This is from last time. And then you would be able to click on the lecture 3 slides, which are the first link, and a link to the recording in the second link. If you go back to announcements, lab materials are there, Zoom link for the lecture is there, the syllabus is there. I don't require a textbook for the course. I give you a free textbook. Textbook is there, etc. So I would recommend to read over the syllabus. Yeah. Okay, good. So at least you know where to find things. No worries, no worries. I'm just happy to know that you know where they are. So yeah, look things over. If you have any questions, send me an email. And if you want to meet for office hours, let me know as well in an email. Okay. All right, guys, I'm gonna head out. Have a good rest of your day, and I will see you on Thursday.